Hi, this is Mr. McLean, and in this session we're going to spend a little time reviewing what's classified as big idea number 2, which focuses in on the concept of using free energy in biological systems. So we're going to talk about different categories of reactions and try to evaluate whether or not they are spontaneous, non-spontaneous, what kind of energy change is going to take place. Is it something that's going to have a positive or a negative free energy change? And hopefully we can get some biological examples into this system here. So when we start off in section 1 of chapter 8, the first concept that really comes across is metabolism. And that really sets us up for the next two chapters that follow, where we look at photosynthesis and we look at cellular respiration. And really throughout the course, we start to see that type of process. So when we look at metabolism, one of the first things that should jump out to us is that almost every reaction that we could potentially look at in a biological system is going to be enzyme catalyzed. So this is a very basic, nondescript progression that we can see. But as we move from an initial molecule and we make our way to product, we know that rarely is it a one-step process. Typically, you're going to have multiple intermediates, in this case, substance B and C, in order to lead us to section D, in order for that to take place. And each one of these involves a separate reaction. The other really, really important thing about chapter 8 is we start to look at the function of enzymes. So we're going to talk about that in a later video, but if I were to knock out enzyme number 2, so let's suppose that there was some type of a genetic defect that didn't allow us to produce enzyme number 2, which is most likely protein-based, then our reaction would be terminated at this particular point. We would not be able to move from B to C to D. So that's a really big idea, not only for this chapter, but also when we start getting into genetics. So getting into the two different areas of metabolic pathways, we're basically going to see there's two general scenarios. Scenario number 1 is we end up with something that is classified as being a catabolic pathway. Okay? And when you think about catabolic, you're talking about releasing energy. Okay? And you're going to break something that's more complex down into something that's going to be simpler. So one of the processes that we'll get a lot of detail on in coming weeks is cell respiration. And if you think about it, when you look at the respiration process, you start off with this glucose molecule. So you got your C6H12O6. And, of course, you're going to have to throw in oxygen into that system. All right. And when you do that breakdown, what we're going to end up with is we're going to get our carbon dioxide, and we're going to get our water. Now, if you compare this molecule of glucose with these two molecules of carbon dioxide and water, it would be fairly obvious that this is a more complex molecule. Okay, so it's going to be a bigger, more complex molecule as opposed to carbon dioxide and water. So whenever you're taking something and you're breaking something that's more complex or larger down into smaller steps, that would be an example of catabolism or a catabolic process. Now, if we want to go in the opposite direction, we have anabolic pathways. So anabolic pathways are going to build complex molecules from simple things, but they require energy. So if you think about it, anytime you're taking something simple, so we could look at the example of building the primary structure of a protein. And we know that the primary structure of a protein is going to rely on a very specific sequence of amino acids. And as we put amino acid number 1 and number 2 and number 3 and number 4, and we use these peptide bonds here, that is what we would classify as being a anabolic process. So we're starting off simple with the amino acids, and we're building something that's going to be more complex. And in order to do that, we're going to have to invest energy. So things that are anabolic are going to require energy. So the whole concept of this unit deals with energy, and we need to think of energy as something that causes, or at least is the capacity to cause change. And of course, we know that there's different forms of energy. You can review some of these different things, but as far as this class is concerned, we talk a lot about heat, because we think about that in many ways as being a type of energy that is it's necessary to keep our homeostatic level in terms of body temperature, but a lot of times we also think of it as an energy source that doesn't directly do work for us. And then we've got chemical energy, which is the energy that's going to be stored and released during a chemical reaction. So one of the pictures that we looked at in class that doesn't necessarily seem like it directly relates to biology, but we can make this application, is with all reactions, what we're going to find is that there are two different scenarios for a reaction. And if we graph this out, you're definitely going to see this type of graph on the test. If we're graphing free energy, okay, so we're just going to say G, which stands for our free energy. And we'll do another graph right here, G for free energy. And we graph that versus time, okay? If we start off with our reactants at a certain energy level, and of course we have to agitate that enough to overcome the activation energy, and we end up with, so here's our reactants, and here's my products. If we look at scenario A versus scenario B, and in scenario B, we do this. But now we're going to look at it coming out at a higher level. These two reactions and graphs of the reactions can correlate with the two things that we just talked about. So if we look at scenario A, what we're going to see is that we're taking our reactants, which have a certain amount of free energy associated with them, energy to do work, and of course, we have to agitate it a little bit, which is our activation energy. We'll call this E sub A. I'll talk about that a little bit more in another video. But after the reaction actually happens, we get to that transition point, we find that we get products that are going to have this much less energy associated with it. So if you get something that does that, that means that energy had to be released from the system. That also means that we have something that is spontaneous. So this is an example of a negative delta G, and we'll talk about the formula here in a minute, but this one is definitely going to be a spontaneous reaction. If we look over here at this guy, if the guy is standing right here at this high point, and I know he's on this platform out in the middle of nowhere, there is a certain amount of instability with being at that particular position. Much like these reactants have a certain amount of instability. They naturally would like to progress to a more stable configuration. So when this guy has his head just above water and he's sort of like swimming in there, once he hits that point, he's at a point of greater stability. If you want to take him and put him back up here, that would be kind of like us going from this point and trying to get us to this point. Okay, so this one right here is going to require an investment of energy. Okay, so we're going to have to invest energy, much like we're going to have to invest energy to take our guy from the water and put him up on top of the platform. So the picture, the graph on the bottom half is one in which energy has to come in, and as a result of that, this is non-spontaneous. Okay, non-spontaneous. Now, there's a couple other factors that we definitely need to look at to make sure that we're good on that, but what we want to focus on is a cellular environment. Now, a couple things to keep in mind. What we're talking about deal very, very closely with the concepts of the laws of thermodynamics. And when you deal with thermodynamics, you have to make sure you know what kind of system you have. So if you have what's classified as a closed system, that would be like the best thermos that's ever been made. Okay, it's supposed to be isolated from the surroundings. And there's really nothing that's going to be 100% isolated, but you kind of get the concept. Cells are not closed systems. We are open, and that is because all organisms rely on taking energy from the environment. They exchange with the organism, and we release different forms of energy out to the environment. And the idea is that we're never in equilibrium in regards to our energy changing. So anything that's alive is constantly going to be moving, trying to achieve equilibrium, but not actually getting to that particular point. All right. If we go over here, we focus on the first law of thermodynamics. The first law is basically that you can't create or destroy energy. Okay, that's sometimes referred to as conservation of energy. So all we're doing is changing forms, but we're not creating energy. The second law is a little bit more confusing for many of us, and that is every energy transfer or transformation is going to increase entropy. Okay, entropy is defined as the disorder of the universe. So there's a lot of different ways in which we can do this, but let's look at this first scenario. We have our predator. We looked at this image in class. We have our predator, and they're getting a food source, and they're not creating energy, but they're transferring it from one form to the next. So now, within that food source, there were glucose, there were different other types of sugars, there were fats, there's proteins. So now, as our predator is going through respiration, what are they doing? They're breaking this down. So if we go in there and we take a look back at the very beginning when we talked about respiration as being a catabolic process, the idea is we took something that was a little bit more ordered, the proteins and the complex carbohydrates and the fats, and we created something with less order. Okay. Another way of thinking about it, if you have something like a stick of dynamite, okay, and the dynamite has components in that molecular structure that gives it a lot of energy, okay? A lot of potential energy, I guess you could say, the way the bonds are arranged. It doesn't do anything unless we overcome that necessary activation energy. But once we get that activation energy achieved and we get to that point of transition, what we're going to find is that our substance that is contained in a very small area is going to have a release in many different directions. Okay. Anytime we look at something that's breaking down or degrading, we're talking about something that's in line with that. All right. Now, one of the things that we find is a lot of people look at it and they say, well, cells are very ordered. Living things are ordered. And that's true. The concept of entropy is that you can have individual components of the universe, that would be living things or cells, that actually become more ordered as long as the total sum of all the processes that are needed in order to make that change lead to greater entropy in the universe. Okay, so it's a little bit of a concept that is sometimes difficult to follow there, but we understand, if you think about even energy transformation starting with producers, you're going to have energy flowing into an ecosystem in the form of light, exiting in a very random form, which is heat. So the idea is that the organisms and cells in general make order from less ordered materials, because we obviously involve, smaller components to become more complex, but putting those things together, the sum total of all of those transformations leaves the universe with less ordered forms of energy. Okay? And that, like I said, that's a little bit of a difficult thing to follow there. So that kind of highlights that right there in this statement. The decrease of an organism is okay as long as the sum total of everything that's going on across the universe leads us to a greater amount of entropy. Okay, I'm going to stop there. I'll put out a second video for section 2. We'll look at a few more of those graphs and hopefully answer some questions.